Welcome to Let's Get to the Marks, going through AQA GCSE Combined Science Trilogy paper. This is the Biology Paper 1, higher, and this was taken on Tuesday the 16th of May 2023. This is a walkthrough, so for each question, pause the video, grab some paper and a pen and a calculator, have a go at the question, then unpause and watch the walkthrough. Which disease is a non-communicable disease? Well, AIDS is a viral disease that you can catch through sexual intercourse or sharing needles. Cancer is not, you're not able to catch, so that is the non-communicable disease. Gonorrhea, caused by a bacteria, sexually transmitted disease, and malaria is a protist that is carried by mosquitoes and so can be spread. 1.2, give one non-communicable disease that obesity is a risk factor for. So if you're overweight and you're classed as obese, what diseases can you get, or is that a risk factor that it contributes towards you getting? And the answer is diabetes. We can't refer to cancer because that was in the previous question and it tells us don't use the word cancer. It says do not refer to diseases given in 1.1. Coronary heart disease or a heart attack or coronary vascular disease, arthritis. These are all possibilities. There are many more, but we'll leave it there. Let's go on to 1.3. National policies are used to help people who are obese to lose weight. One national policy is to reduce the amount of sugar added to foods and drinks. You've seen they've got a sugar tax at the moment in the UK, where actually all companies that produce foods with sugar in have to pay a tax. So another way the government could encourage people to lose weight is to encourage exercise. They could do this through education. They could make a law making everybody exercise. That would be a bit extreme, but they could do that. They could give free gym memberships to people who are obese so that they're encouraged to lose weight. They could offer discounts on people that lose weight or discounts on healthy food, make healthy food cheaper. So there's all sorts of things they could do to encourage people to lose weight, and any sensible suggestion will score you the mark here. So there's hundreds of possible answers there. More PE lessons for kids in school, diet plans given out by the GP to people who are obese or overweight. They probably do that now anyway. All right, let's move on to 1.4. So 1.4, the body mass index, known as the BMI, is one measure of obesity. BMI is calculated using the following equation. BMI equals body mass in kilograms divided by your height in meters. Table 1 shows how BMI is used to describe an adult's BMI category. So BMI below 18.5, you're underweight, 18 to 24, you're heavyweight, 25 to 29.9, you're overweight. A person who is 1.64 meters tall and has a mass of 69 kilos, determine their BMI category for this person. Use the BMI equation in table 1 and it's worth 3 marks. So we want to do the kilograms over their height squared. So 69 kg over 1.64 squared. Remember to bracket the 1.64 squared. You should really put the squares inside the brackets. And then when you get your answer, look it up in the table and see which range it falls in between. So if you bust that into your calculator, you should get something like 25.65, something along those lines, and you will have scored 2 marks for that. Now you need to select the correct range. Are they in heavyweight? No, that only goes up to 24.9. They're between 25 and 29, that answer is. So they are overweight, and that gets you the 3rd mark for getting the correct category. Okay, let's zing down to 1.5. Scientists investigated the effect of smoking and of BMI on the birth mass of babies. Women's BMI categories were determined before the women became pregnant. 1.5 suggests why BMI categories were determined before the women became pregnant. Why is it important to test women's BMI before they're pregnant? Think about what happens to one of those values in the BMI equation when you get pregnant. Well, obviously, when you're pregnant, you gain mass, you get heavier, your weight increases. And is that fair to judge someone's BMI when you're weighing the mass of a pregnant person? That means you're also weighing the mass of the baby or the developing baby called the fetus. So you're not really getting the woman's BMI, you're getting her BMI and her baby's mass included. So it's not really the woman's BMI, so to speak. So it's not really a fair evaluation of the person's BMI. So it's better to measure a woman's BMI before they become pregnant. So if we look at figure one here, it shows the mean birth mass of different of babies when people are non-smokers or smokers, or women are non-smokers or smokers. Give two conclusions that can be made from figure one. Now, conclusion, you need to know your command words, just means two things you can state from the evidence that are absolutely true facts. You don't need to explain using any scientific knowledge. So what two facts can we say from this data? So, for example, at all weight categories, non smokers' babies had a higher birth mass than smokers. Or we could say the opposite and say smokers' babies always have a lower birth mass than non smokers in the same weight category. So that's a fact, right? We could also say that obese non smokers have the babies with the biggest birth mass, and we could also say that underweight smokers have babies with the smallest birth mass. So there's literally a whole range of answers you could give here. These are just two of them or three of them that I've given here. You could pick any two of these three, but there's 10 or 15 things you could say that are factual, two conclusions you can draw from this graph, nice and easy from a whole range of different possible answers. All right, let's move on to the next question. So 1.7, measles is a communicable disease. A virus causes measles. Describe how the measles virus is transferred from person to person. So how do you catch measles? It's when people cough and droplets in the air spread. And people breathe those droplets in, or they land on their hands and they touch their face, or those droplets hit door handles and you touch the door handles and then your mouth. Okay, 1.8. Athlete's foot is a communicable disease. A fungus causes athlete's foot. The athlete's foot fungus infects the skin on the feet. 1.8. Scientists estimate that 17% of the UK population have got athlete's foot. The estimated UK population is 67,961,900. Calculate how many people are estimated to have athlete's foot. 2 marks. So here we have a mental maths. One is just basically doing percentages. Can you work out what 17% of 69,961,900 is? So 17 over 100, or you can just in your head 0.017 times 67,961,900, and you're going to get your answer, which is something along the lines of 11,553,523 people have athlete's foot. That's crazy. That's crazy. Nearly a fifth of the population have got fungus on their feet. 1.9, athlete's foot grows in moist conditions, suggests one way a person could reduce their chance of getting athlete's foot. So how could you stop yourself getting athlete's foot? Obviously, the fungi want sugar, they want water, so they want moisture. So just make sure you dry your feet after a shower properly in between the toes. You don't share towels with other people because they might have athlete's foot and you might pick it up off them. And you shouldn't walk bare feet in the swimming pool, in the changing rooms. And really, walking around barefoot is an easy way for you to catch athlete's foot of someone else if you're on a hard surface. You can use a fungicide, sprays, creams, or powders on your feet that will kill any fungus. And wear socks if you're in a public place, like you're walking around your friend's house. At least keep your socks on your feet, and then you won't pick up their athlete's foot if they have it. And that's the end of question one. Let's move on. Keep moving on, keep moving on, keep moving on, keep moving on. Question two. Figure 2 shows onion cells viewed using a light microscope. We've got a field of view here in this circle, times 100 mag. Figure 3 shows the apparatus given to a student, microscope, 15 centimeter ruler, and a prepared slide with onion cells on. That means it's got the cover slip on the glass slide and a little drop of water in there. Describe how the student could use the apparatus to estimate the mean length of onion cells in the slide. So I'm going to go back up to the equipment and we'll have a little think. Now, there's a couple of ways to do this, and there's like 3 different methods in the mark scheme. I'm just going to go through one of the methods that you can use to work out the mean length of one of those cells. So let's get into it. First of all, you can put the slide on the stage, clip it in. Obviously, it's already been stained and a cover slip put on it and a droplet of water so the onion cells are floating in water. You don't need to add any of that because it told us the slide is prepared. I place the ruler next to the slide. Now, use the ruler to measure the size of the field of view. You're going to want to get the field of view in focus so you can see all the cells. And once you can see the cells, measure how long the whole field of view is with the ruler. And you want to measure that in millimeters. Next, count how many onion cells appear in the field of view. And I'll literally draw this on here so you can see what I mean. So what we want to do, you can go across or down, it doesn't matter as long as you know which way you've measured. So look, 1, 2, 3, 4, 5, 6, 7. Right, I've got 7 cells going down in my field of view. That's how many cells fit in the field of view. Now, if I've already measured that using the ruler and I know that distance in millimeters, I now just need to divide the length of that in millimeters by the number of cells in the field of view. Okay, next question. The circulatory system includes the heart, the blood vessels, and blood. The heart pumps the blood in a double circulatory system. Describe what is meant by a double circulatory system. Check out my grade D GCSE art over here. I've got the right atrium and the right ventricle, and I've got a blood vessel carrying the blue deoxygenated blood up to the lungs. So, why is it a double circulatory system? Well, one of the answers is because the right side of the heart or the right ventricle pumps blood or deoxygenated blood to the lungs. And that's one of the circulations we've got. Now, the second circulation is that the left side of the heart or the left ventricle pumps oxygenated blood round to the rest of the body. So here is your left atrium and left ventricle on my crusty diagram. Here's the red oxygenated blood coming in back in through the left atrium, down to the left ventricle, pumped up round the back of the heart and off round to the body. And so we have a double circulatory system. Essentially, the right side pumps blood to the lungs, the left side pumps blood to the body, and that would be good enough for you to get 2 marks in simple terms. If you want, you can mention that the right side's deoxygenated and the left side's oxygenated. Right, 3.2. Heart rate is controlled by a group of cells that act as a pacemaker. Where in the heart is the pacemaker found? So, which of these chambers, can you remember, do we find the pacemaker? It's actually on the right side and it's in the wall. It's here where this arrow is. It's inside the wall of the right atrium. And you need to remember that. That's a fact that's come up a number of times, a little 1 marker. So not the end of your exam if you don't get it right. Right, let's zoom in on 3.3. Which blood vessel carries deoxygenated blood? So let's take a look over here at the heart, and the deoxygenated blood enters the lungs through one blood vessel, a big one called the vena cava. It goes into the ventricle and then it comes up this tube to the lungs. So it enters the heart via the vena cava and then travels up a second tube, the deoxygenated blood, to the lungs. And that's the pulmonary artery, the only artery in the body that carries deoxygenated blood. How do we know it's an artery and not a vein? If you're going away from the heart, begin with an A. Away, you're an artery. Artery and away both begin with an A. 3.4, explain why veins have valves but arteries do not. So we have to think about it. Why do veins need valves? Well, veins are carrying blood at a lower pressure. Therefore, there's a tendency for that blood to maybe fall backwards. And so we need valves to prevent the backflow of blood. Arteries are being squeezed. Imagine like you're squeezing a fairy liquid bottle or a water bottle. Arteries are being squeezed under high pressure. They don't need valves. Then the blood is not going to flow in the wrong direction with arteries. It's being squeezed at high pressure. 3.5, describe 2 structural differences between a vein and an artery. So do not refer to valves because we've already talked about them. So here is my vein over on the right and my artery on the left. What 2 physical differences can you notice other than my little dotted valves? Okay, so first of all, the artery has a thicker wall. You can say, if you want to be more technical, it has a thicker layer of muscle and elastic. So veins also have elastic and muscle in their wall, but arteries have more muscle and more elastic because they're carrying blood at higher pressure, bigger contraction, and elastic is to smooth the flow of blood and to cope with recoil. Now, arteries have a smaller lumen. That's the space in the middle, the circle in the middle. You can see I'm drawing a red line to show the diameter of the lumen. Veins have a larger lumen. They're carrying blood at a lower pressure. Okay, let's move on to the next question. This is question 4. A leaf is a plant organ. Figure 4 shows the tissues in the leaf. We've got 4 different tissues, A, B, C, and D. Which tissue is the epidermis? Now, epi means outer. So which of these, A, B, C, or D, is on the outer layer? And the epidermis, the outer layer, is going to be A. And it's made up of upper epidermal cells. They happen to be transparent to let light through if you needed to be asked about that, and also covered in a waxy cuticle to waterproof and reduce water loss. You also find the epidermis at the bottom of the leaf as well, facing the outside. Explain how the spongy mesophyll is adapted. So look at this layer in the middle here. This is called the spongy mesophyll. You should notice where this black arrow is pointing, we've got big air gaps, okay? We've got large air gaps. And what do these large air gaps do? Well, they allow a certain gas in and a certain gas out, and they allow for rapid diffusion of gases through the leaf tissue. So the gas that we want coming into plants is carbon dioxide, because you need carbon in glucose, and the gas that's going out is the waste product of photosynthesis, which is oxygen. Hence, we call it gas exchange. So having air gaps allows those gases to rapidly get to the cells they need to. Carbon dioxide needs to get to tissue B, which is the palisade cells. Those palisade cells are where all the chloroplasts are and where the carbon dioxide is needed for photosynthesis to make glucose. So explain how the spongy mesophyll is adapted to its function. It has large air gaps. These allow gas exchange, rapid diffusion of carbon dioxide over to the palisade cells. You can call them palisade mesophyll cells if you want. It also allows the oxygen produced in the palisade mesophyll cells, which is a waste gas, to be rapidly diffused out through the air gaps and out of the stomata. Okay, we're going to get us 3 marks for that answer there. 4.3, the xylem is adapted to transport water through a leaf. Explain one way the xylem is adapted to its function. So you should only give one way. They've strictly said just talk about one. They're going to ignore it if you talk about 2 or 3. So it contains lignin, which is a chemical that makes the walls of the xylem strong. Now it's carrying water under a big pressure, and this stops the walls breaking or bursting. It also helps keep the plant upright. Now, another possible answer you could have gone for, it has no cell structures, and you could have named one. So it doesn't have mitochondria, the xylem doesn't have cytoplasm, it doesn't have a nucleus. So you could name any cell structure, no ribosomes, and that means there's less resistance to the flow of water, or you could just say it's easier for water to flow up the plant because there are no obstacles. 4.4, figure 5 shows a pair of guard cells around a stoma. That's a singular for stomata, the hole in the middle. The thinner part of the cell wall's on the out, and the thicker part of the cell wall's on the inside near the hole, the stoma. During the day, glucose is made in the guard cells. Those are orange circles I've made. That's the glucose. Describe how an increase in glucose concentration in the guard cells causes the stoma to open. If you put sugar inside a cell or glucose inside a cell, you lower the water potential or the water concentration. You make the solution more concentrated and less watery. This means that water moves in from the surrounding cells into the guard cells by osmosis, makes them swell up, and it makes that hole in the middle bigger. Okay, so quite simply, the water potential in the guard cells is lowered. Water moves into the guard cells, causes the stoma to get bigger as the guard cells swell up. The thicker inner wall curves around the stoma. And if you're ever asked, how does this mechanism work? It's because it has a thin wall on the outside and a thick wall on the inside. It's about all you got to remember. Okay, we've got another six marker here. So, light intensity varies during 24 hours. Figure 6 shows the overall exchange of carbon dioxide and oxygen for a tree at three different light intensities. This was one people found really difficult in the GCSE when it came out. We've got an arrow going in at bright light and an arrow coming out at bright light. So, during bright light, we're thinking about photosynthesis. So, probably carbon dioxide in, oxygen out. Yeah, so the oxygen's going out. And then we're looking at when there's no light, there's probably no photosynthesis happening when there's no light, right? So we're probably looking at oxygen still going in, because what is the plant doing? There's another chemical reaction a plant is doing, not just photosynthesis. All living things do this reaction, which requires oxygen and produces carbon dioxide. And that's happening when there's no light. And then we've got dim light with no arrows in or out. That's interesting and probably the hardest thing to work out what's going on there. There's no overall CO2 or oxygen going out. So what must be going on in the dim light? So we've got in bright light, the plant is doing lots of photosynthesis, which produces some oxygen. So we've got some oxygen coming out. Of course, the plant is still respiring. So some of that oxygen is used up. And the plant is doing lots of photosynthesis, which uses CO2. So we've got a lot of CO2 being brought in, shown by the solid black line, lots of CO2 coming in and being absorbed from the air. The CO2 made by respiration is used by the plant um, for photosynthesis, so we don't really get any CO2 coming out. Um, and there is some oxygen produced by photosynthesis, and some of that will be used in respiration. But overall, there might be a little bit more oxygen going out because we've got more photosynthesis than respiration happening in bright light. So, photosynthesis is happening at a higher rate than respiration in the bright light. But this was a difficult question. We've then got the dim light. If we look at the dim light next and what's going on in the actual dim light, well, there's no arrows in or out. Does that mean the plant is not taking in carbon dioxide or taking in oxygen? Of course not. In the dim light, the rate of respiration and photosynthesis are equal. So the amount of CO2 going in for photosynthesis is exactly equal to the amount of CO2 being released by respiration. The amount of oxygen coming in for respiration is exactly equal to the amount of oxygen going out, which is produced in photosynthesis. So the two rates are the same. So the products and reactants being produced are the same in dim light. We've got no overall arrow going in or out. And then for the final bit, in no light, of course, you get no photosynthesis, which means no CO2 is going in, but you still got some CO2 going out. Why is that? Well, the plant, although it can't do photosynthesis, it can still do respiration. Its mitochondria can still carry out respiration. It doesn't require light for respiration. So it's still producing CO2, is why we've got a solid black arrow. A lot of CO2 is being released. It's taking a little bit of oxygen in, obviously, for respiration, and it's not really producing any oxygen by photosynthesis. So it's having to get all of its oxygen from the atmosphere at night. And there we go. That's another six marker. So let's move on and check out the next question. And we should be well over halfway through this paper now. Don't forget to hit the like and subscribe, and make sure you share, because by you helping me, I can help you by doing more of these papers, basically to help you smash the exams. All right, let's move on. Question 6. Amylase is an enzyme that digests starch in the digestive system. Explain why starch has to be digested. Okay, so starch is insoluble. It doesn't dissolve. You know that when you wash rice and the water goes cloudy, or if you make some pasta and the water's cloudy after, that's the starch molecules floating in the water. Starch is insoluble. So it can't be transported in the blood because it's insoluble. It's large and it needs to be broken down so it can be absorbed and transported in the blood, essentially. So that's why we have to digest starch. Starch can be digested into glucose, which is a soluble monomer of the insoluble polymer that is starch. And then once it's in glucose form, it can be transferred around our body in the blood and taken to any tissue or cell that requires energy by respiration, which is every single one of them. A student used a colorimeter to investigate the rate of starch digestion. A colorimeter measures the percentage of light passing through a liquid. The darker the color of the liquid, the less light passes through. The student mixed 1 centimeter cubed of starch suspension with 10 centimeter cubed of iodine solution, measured the percentage of light passing through the mixture, and repeated with different concentrations of starch suspension. Figure 7 shows the results. We have the percentage of light on the y-axis, the percentage of light passing through the mixture, and on the x-axis we have the concentration of starch in grams per decimeter. 6.2 suggests what liquid was used for the test when we needed 0 gram per decimeter of starch concentration. Of course, what are you going to use if you want something with no starch? You're going to use distilled or deionized water. You're just going to use water. Normal water doesn't have any starch. 6.3, explain the change in the percentage of light through the different concentrations of starch suspension in iodine solution. Notice I've circled explain, that's the command word. It means don't just describe it. You will get 1 mark for describing, but the other 2 marks are for using science. So we need to zing in here and say what is happening to the percentage of light passing through the mixture as we increase the concentration of starch. So the higher the concentration of starch, the lower the percentage of light passing through the mixture. Why is that? Well, when starch mixes with iodine, it goes blue or black. And that means if you have more starch, you're going to get a darker solution, a blacker mixture, or a dark blue. It's like blue stroke black, so it's like blue black. So you're going to get a more blacker solution or a more bluer solution the more starch you use. And therefore, that explains the pattern. So what was the first mark? The first mark was just to say, as the starch concentration increases, percentage of light passing through the mixture decreases, less light passes through. This is because as we increase the starch concentration, the mixture is darker because it goes blue-black with starch, so less light can get through, more light is absorbed by the blue-black mixture, and that is the 3 marks. Boom! The student then investigated the rate of starch digestion using amylase. This is the method they used. 10 centimeter cubed of starch suspension in a test tube. Do you know what? I'm not going to read it all. Just press pause on the video and read through this method. You'll see they've used 37 degrees for the water bath. They did the test for 10 minutes. They put them both in for 10 minutes and allowed them to equilibrate. We'll look at why that is in a second. Then they removed 1 centimeter cubed of the mixture and added it to the 10 centimeters of iodine solution and measured the percentage absorption. Right. 6.4, the starch suspension and the amylase solution were kept in the water bath for 10 minutes together before being mixed. Why? Well, think about it. They've both got different specific heat capacities. They might be both at 2 different starting temperatures. We want them both to get to 37 degrees or the optimum temperature. So we set the water bath to 37 degrees and we want them both to get there before we start. Percentage of light passing through the mixture, we can see here it's increasing over time in minutes. So what's happening here? It's the total opposite of the graph we looked at originally, graph 7. So what is happening over time to the amount of starch? As starch increased, less light got through because it got darker. But here, something is happening to the starch. And of course, you know the amylase is breaking the starch down, hydrolyzing it or catalyzing it into glucose. So instead of it going dark blue, it's actually getting lighter as the starch gets broken down. Now we've got a difficult little calculation. The concentration of starch at 3 minutes was 0.3 gram per decimeter. Calculate the mean rate of starch digestion for the first 3 minutes. Use figures 7 and 8. Now this is the hardest question in the paper, so we're going to get into it. Okay, so we need to look at both figure 7 and figure 8 to solve this one. I'm going to start with figure 8. We want to find out what the percentage of light was when we just had water in there, when there was no starch. Sorry, not when there was water. We want to find out the percentage of light when the starch was first put in and none of it had been broken down. So it looks like when it was just pure starch and it was obviously dark blue, only 10% of light got through. So now we need to know, well, what's the concentration at the start? Because if we're going to do the rate, we need the initial and final concentration. So the initial concentration at 10% was, we're going to need to go to figure 7, find 10% on figure 7, it's right here. We know that the starch concentration was only letting through 10% of the light, and so we draw a line across from here to the curved line, and we will know what the concentration of starch is when only 10% of light gets through. So it's obviously quite a high concentration because not a lot of the light is getting through, and that's because the starch makes the iodine blue black and absorbs a lot of the light. So look here, it goes right past 6, 3 little squares past 6, and that works out on the scale. Each square is like 0.2, so it's 6.6 grams per decimeter of starch. Now we know that 10% was right at the start of the experiment. That's how much light got through at the start in the first 0 seconds. So we know the starch concentration at the start was 6.6. We're told in the stem of the question that the starch concentration at 3 minutes was 0.3, and we're asked to find the rate over that 3-minute period. So we know it ended up it started at 6.6, ended at 0.3. So let's find the difference. Find out how much starch broke down over the 3 minutes. If it was 6.6 and after 3 minutes it was 0.3, how much starch had broken down to become like maltose or glucose? How much was broken down by the amylase? So think about it. 6.6 minus 0.3, it means that 6.3 grams of starch was broken down over those 3 minutes. Now, when we calculate rate, we want to do the volume, the amount, the distance, whatever you're measuring over time. So we're going to do 6.3 grams here, which was broken down over the 3 minutes. 6.3 divided by 3 minutes. We're going to find that there was 6.3 grams per decimeter broken down over 3 minutes, and 6.3 divided by 3 is 2.1. So 2.1 grams per decimeter broken down per minute. So the rate is 2.1 per minute. They gave us the units for the rate, so we didn't need to worry about that. And that would get you all the 4 marks. But this was a particularly hard question and definitely the hardest in the paper. So if you were baffed by this, don't beat yourself up. Okay, 6.6. The investigation was carried out at pH 7. Explain how the results would be different if it was carried out at pH 1. Well, pH 1 is very acidic. Hopefully, you know that's the strongest acid. You do that in chemistry. A much lower pH means more acidic, and it would actually denature this enzyme, amylase. It doesn't work in those acidic conditions. Protease loves it. So pepsin, the enzyme in the stomach, that loves acidic conditions, but amylase doesn't. And this means it would denature. Its active site would no longer fit like a lock and key with the substrate, so it would no longer bind with the substrate. And this means the starch would not get broken down at all. And therefore, what would happen to the absorption of light? Well, it would stay at only 10% of light passing through and 90% of the light getting absorbed. So it would stay at the starting point. Only 10% of the light would get through because the solution would stay just as dark. It wouldn't get any lighter. It would stay just as dark blue because none of the starch could be broken down because the enzyme was denatured, which means the active site had changed and it could no longer fit with the starch and break it down. The starch in this case is the substrate, the amylase is the enzyme, and the active site is where the substrate fits like a lock and key. Right, 7.1. Some strains of bacteria are resistant to antibiotics. Antibiotics are drugs used to treat bacterial infections. Mutations in bacteria produce new strains. Some strains of bacteria are resistant to antibiotics. So we've got to remember that bacteria have no nucleus, but they do have DNA. So a mutation is a change in a gene. So it must happen in a gene or DNA, in fact, in circular or free DNA, or bacteria have tiny little circles just made of a couple of genes called plasmids. So you could say in circular DNA, just DNA, genes, chromosomes, or plasmids. Any of those would be good. Scientists investigated which antibiotics, A, B, C, D, or E, killed Staphylococcus aureus, S. aureus bacteria. This is the method used. So they grew the S. aureus bacteria in a petri dish, cut 5 small discs of paper, soak each disc in a different antibiotic solution, put the 5 discs into the petri dish, keep the petri dish at 37 degrees for 24 hours. Figure 9 shows the results. A clear area around the disc is where the bacteria have been killed. The scientists concluded S. aureus was resistant to antibiotic C and E. What's the evidence? When you look at that picture, what was the evidence it was resistance to C and E? There was no clear area, no white circle around C and E, and this should tell us that no bacteria have been killed. And that's the evidence for 2 marks. So no clear area, bacteria have not been killed. The scientists later discovered that S. aureus is not resistant to antibiotic E, suggest how they changed the method and showed that S. aureus is not actually resistant to antibiotic E. What could have they changed and then suddenly there was a clear area? Well, what they did was they increased the concentration of the antibiotic, made it 2-3 times more stronger. So increased the concentration, and then they found that there was a clear area, which told them that the bacteria were killed and meant that S. aureus isn't resistant to antibiotic E. It's just they weren't using enough of it. So there's our evidence for 2 marks. They increased the concentration, then the bacteria were killed, and a clear area appeared. That's the evidence. Broken bones are sometimes repaired using a metal implant. The area around an implant can become infected with S. aureus bacteria. The infection is usually treated with long-term course of antibiotics. Long-term use of antibiotics has led to the development of antibiotic-resistant bacteria. Research is being carried out into alternative treatments. Stem cell from bone marrow and from fat tissue have antimicrobial properties. The scientists investigated the effect of the 4 treatments on the area of infection around the metal implants. Each treatment was injected into the area around the implant. After 7 days of treatment, the scientists calculated the ratio area of infection against total tissue area. What is the independent variable? Well, what did they change? They changed the type of injection, the type of treatment. Suggests one advantage of using stem cells from fat tissue rather than bones. Do you think it's easier to take some of your fat cells or to get into your bones and take some of those cells? It's obviously easier to remove fat cells. It can be less painful than sticking a needle into your bone and or cutting into your bone and taking some cells out. So less painful, less difficult surgery, easier to get to the fat cells. 7.6. Stem cells containing antibiotic were produced by growing the cells for 24 hours in a solution containing the antibiotic. How did the antibiotic enter the stem cells? Well, if you've put them in a solution of pure antibiotic and then some antibiotic has ended up in the cells, it means they moved in by diffusion, obviously from a high concentration in the solution to a low concentration inside the cells. Yeah, and that will get you 2 marks. So you just really, this question is an application, but it's really just asking you what is diffusion. It's kind of their way of testing you what diffusion is. So by diffusion, it moved from a high concentration outside the cells to a low one in the cells. Right. Now we've got a graph with range bars. So we show the area of infected tissue on the left, and then underneath the 4 different treatments: unreactive solution, antibiotic solution, stem cells only, and stem cells containing antibiotic. You can see here are the plots for all the results of the people they tested it on, or the areas of tissue they tested it on. And you can see that there's a big spread with some of them. What is the range of results for the treatment with stem cells only? So we go to the bar, or on the x-axis, we find stem cells only, and we look. It starts at, I draw a line across to the y-axis, and their first result was at 0.09, and their last result, the last x, if we got with stem cells only, was at 0.31. So the range is from 0.09 to 0.31, and that's what you have to write. You don't need to take one from the other and quote the difference. So it's just 0.09 to 0.31. Now, this is the last part to the question. 7.8. A student looked at the results and concluded injections of stem cells containing antibiotic should be used to treat all implant-related infections. Evaluate the student's conclusion. So this is basically say the positive things and the negative things. When you say evaluate, say the good things about it and the negative things. This is a bit of a silly statement. You shouldn't say treat all infections, because how many different bacteria did they study with these antibiotics? So let's look at the for and against for this one. So zigging back up to the graph, we're looking at the where is that graph? Here it is. So stem cells containing antibiotics. So we're looking at the final column here. The student said this is the best one, and they've definitely got some good reasons to say that one is the best one. So what I would say the pros are is that the results on that one were all very close together. Look here where the 5 little crosses are on the right hand side. This is the stem cell containing antibiotic. That shows that the results were pretty reliable because they're all very close together. And also up on the y-axis, the area of infection, the total amount of infection, this had the smallest area of infection. Look at the range compared to the others. It's a very narrow range, which shows the results were very good. And then across here, when we look here, the area of infected tissue was lowest when we use stem cells with antibiotics. So they're the two good things we can say about it. So let's go and type those in. They're the two best things we can say about this. So the student's kind of right in that it had the smallest range of results, which shows that that it was very accurate and reliable. These results, they're close together. Also, it had the lowest area of infection. So we might say, yeah, this is the best. You're going to get the least area of your body infected if you use this antibiotic. So these are positive things to say. But now we need to come up with at least two negative, and there's a lot of negative things. How many people did they test this on? They only used 5 patients. You need to use this on a lot more patients, maybe 100 or 50 patients. And they only did this over 7 days. If you read the stem of the question, they only did this over 7 days. That's bad. They also say this is for all infections, but they only studied one antibiotic. They only studied one bacteria. So how can they say that this applies to all different infections caused by other bacteria? Also, stem cells can carry a virus. Something you need to know about stem cells is that they can get rejected as well by your body. And antibiotics are a drug, and they can have side effects, or you can have allergic reactions to the antibiotics. Some of the patients might actually be unhealthy. We didn't learn a lot about the patients. We didn't learn about quite a lot of other things about them. We didn't know their body mass. We didn't know their age. We didn't know lots of different things about the patients that could affect these results as well. Okay, guys, sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks. I'll see you guys in the next video.
You're still here? It's over.